take super long today, and still about a half an hour. So we're going to do a video on vision and a video on hearing, just to give you some brief information about vision and hearing to go along with the anatomy that you learned earlier in the week. And then I will briefly try to give you some helpful hints for the 2 handouts that are due tomorrow night. Vision and hearing handouts that go along with visible body. So let's start with this lovely crash course video. Okay, I have a feeling I didn't share audio, so let me try this again. I always forget to click that button at the bottom.
A mess with your brain. This is the first stage of an optical illusion. Many illusions use patterns of light or perspective to exploit the disconnect that exists between sensation and perception, between what your eyes see and what your brain understands. But not all illusions work that way. Some produce ghost effects or after images that take advantage of glitches in the physiology of human vision, like this flag. I'm not trying to make a political statement here, and I'm not going to ask you to swear allegiance to the Republic of Hank or anything. I mean, if I was going to start my own country, my flag would be way cooler than that. Not that I've thought about that a lot. And now look at this white screen. If you looked at that flag for at least 30 seconds without moving your eyes, you'll see something, even though the screen is blank. An after image of the flag. But instead of being turquoise and black and yellow, it's red, white, and blue. Okay, so that's pretty cool, but I'm not here just to entertain you. This kind of illusion is actually a great way to explain your very complex sense of vision. And I do mean complex. Nearly 70% of all the sensory receptors in your whole body are in the eyes. Not only that, in order for you to see, perceive, and recognize something, whether it's a flag or a handsome guy in glasses and a sport coat sitting behind a desk, nearly half of your entire cerebral cortex has to get involved. Vision is considered the dominant sense of humans, and while we can get along without it, and it can be tricked, what you are about to learn is not an illusion. When we talked about your sense of hearing, we began with the mechanics of sound. So before we get to how your eyeballs work, it makes sense to talk about what they're actually seeing. Light bouncing off of stuff. Light is electromagnetic radiation traveling in waves. Remember how the pitch and loudness of a sound is determined by the frequency and amplitude of its wave? Well, it's kind of similar with light, except that the frequency of a light wave determines its hue, while the amplitude relates to its brightness. We register short waves at high frequencies as bluish colors, while long, low frequencies look reddish to us. Meanwhile, that red might appear dull and muted if the wave is moving at a lower amplitude, but super bright if the wave has greater amplitude and thus higher intensity. But the visible light we're able to see is only a tiny chunk of the full electromagnetic spectrum, which is from short gamma and X-rays all the way to long radio waves. Just as the ear's mechanoreceptors or the tongue's chemoreceptors convert sounds and chemicals into action potentials, so too do your eye's photoreceptors convert light energy into nerve impulses that the brain can understand. To figure out how all this works, let's start with understanding some eye anatomy. Some of the first things you'll notice around your average pair of eyes are all the outer accessories, like the eyebrows that help keep the sweat away if you forgot your headband at racquetball, and the super sensitive eyelashes that trigger reflexive blinking, like if you're on a sandy beach in a windstorm. These features, along with the eyelids and tear-producing lacrimal apparatus, are there to help protect your fragile eyeballs. The eyeball itself is irregularly spherical with an adult diameter of about 2.5 centimeters. It's essentially hollow, full of fluids that help it keep its shape, and you can really only see about the anterior sixth of the whole ball. The rest of it is tucked into a pocket of protective fat, tethered down by 6 strap-like extrinsic eye muscles and jammed into the bony orbit of your skull. While all this gear generally does a fantastic job of keeping your eyeballs inside of your head, which is good, On very rare occasions, perhaps after head trauma, or even a really intense sneeze, those suckers can pop right out, a condition called globe luxation, which you really do not want to Google. I'll just sit here while you Google it. Now, you don't need to pop your eyeball out in order to understand how it's structured. I'll save you the trouble and tell you that its wall is made up of 3 distinct layers: the fibrous, vascular, and inner layers. The outermost fibrous layer is made up of connective tissue. Most of it is that white stuff called the sclera, while the most anterior part is the transparent cornea. The cornea is like the window that lets light into the eye, and if you've ever experienced the excruciating pain of a scratched one, you know how terrible it can be to damage something so loaded with pain receptors. Going down a little deeper, the wall's middle vascular layer contains the posterior choroid, a membrane that supplies all of the layers with blood. In the anterior, there's also the ciliary body, a ring of muscle tissue that surrounds the lens, but the most famous part of this middle layer is the iris. The iris is that distinctive colored part of the eye that is uniquely yours. It's made up of smooth muscle tissue shaped like a flattened donut and sandwiched between the cornea and the lens. Those circular sphincter muscles, yeah, that's right, you got sphincters everywhere, contract and expand, changing the size of the dark dot of your pupil. The pupil itself is just the opening in the iris that allows light to travel into the eye. You can see how an iris protects the eye from taking too much light in. If you shine a flashlight in your friend's eye in a dark room, their pupils will go from dilated to pinpoints in a couple of seconds. Light comes in through the cornea and pupil and hits the lens, the convex transparent disc that focuses that light and projects it onto the retina, which makes up the inner layer of the back of the eyeball. Your retinas are loaded with millions of photoreceptors, which do the crucial work of converting light energy into the electrical signals that your brain will receive. These receptor cells come in 2 flavors, rods and cones, which we'll back to in a minute. But the retina itself has 2 layers: the outer pigmented layer that helps absorb light so it doesn't scatter around the eyeball, and the inner neural layer. And this layer, as the name indicates, contains neurons. Not only the photoreceptors, but also bipolar neurons and ganglion neurons. These 2 kinds of nerve cells combine to produce a sort of pathway for light, or at least data about light. Bipolar neurons have synapses at both ends, forming a kind of bridge. On one end, it synapses with a photoreceptor, and at the other, it synapses with a ganglionic neuron, which goes on to form the optic nerve. So say you've just been hit with a blinding flashlight beam. That light hits your posterior retina and spreads from the photoreceptors to the bipolar cells just beneath them to the innermost ganglion cells, where they then generate action potentials. The axons of all those ganglion cells weave together, create the thick, ropey optic nerve, your second cranial nerve, which leaves the back of your eyeball and carries those impulses up to the thalamus and then onto the brain's visual cortex. So that's the basic anatomy and event sequencing of human vision. But what I really want to talk about are those 2 types of photoreceptors, your rods and your cones. Cones sit near the retina's center and they detect fine detail and color. They can be divided into red, green, and blue sensitive types based on how they respond to different types of light. But they're not very sensitive, and they really only hit their activation thresholds in bright conditions. Rods, on the other hand, are more numerous and more light sensitive, but they can't pick up real color. Instead, they only register a grayscale of black and white. They hang out around the edges of your retinas and rule your peripheral vision. Since these receptors function so differently, you might not be surprised to learn that your rods and your cones are also wired to your retinas in different ways. As many as 100 different rods may connect to a single ganglion cell, but because they all send their information to the ganglion at once, the brain can't tell which individual rods have been activated. That's why they're not very good at providing detailed images. All they can really do is give you information about objects' general shape or whether it's light or dark. Each cone, by contrast, gets its own personal ganglion cell to hook up with, which allows for very detailed color vision, at least if conditions are bright enough. And all this brings us back to that weird flag. Why does staring at this flag and then looking at an empty white space make us see a phantom flag of different colors? Well, it begins with the fact that our photoreceptors can make us see after images. Some stimuli, like really brilliant colors or really bright lights, are so strong that our photoreceptors will continue firing action potentials even after we close our eyes or look away. The other part of the illusion has to do with another bug in our visual programming, and it's just that our cones can get tired. If you stare long enough at a brightly colored image, your cones will receive the same stimulus for too long, basically stop responding. In the case of the flag, you looked at an image with bright turquoise stripes. Because your retinas contain green, red, and blue sensitive cones, the blue and green ones got tired after a while leaving only the red cones left to fire. Then you looked at a white screen. That white light included all of the colors and wavelengths of visible light, so your eyes were still receiving red, green, and blue light, but only the red cones were able to respond. As a result, when the afterimage began to appear, those stripes looked red. The same thing happened to your rods, except since they only register black and white, the afterimage was like looking at a negative of a photograph, dark replaced with light. That's how those black stars and stripes turned white. So yes, human vision is fallible, but those mistakes that it makes can help us understand that wonderfully complex system. And that
Okay, so that gives you a nice brief overview of vision and a little more visual functional stuff than just our anatomy we were doing the last few days. So now I'm going to pull up the assignment. It's on Blackboard. So you have this handout. So just to help you figure it out and some of the quirks. Well, I've made underlines here because this is originally a handwritten assignment that's turned in, but I kept it as a Word file, changed it from PDF to a Word file. So there's always those quirks. But of course, you can always delete the lines. I probably should just left them gaps. Lab section is either A or D. Most of you are in lab section A. Your name is the most important part. So just type in here your name. Then, because it's a PDF file, of course, usually it's designed for you to write on these lines. I converted it to Word and added text boxes. So if you click next to it, you should get the text boxes to come up so you can type in your answers here. If you have a good way to handwrite on these lines, you can do it. I just would prefer you not, because the nice thing about typing this stuff up is I don't have to worry about interpreting people's handwriting. My handwriting is terrible. I am not criticizing, just typing is much easier to read. So some of it's anatomy. Then you'll go through and I'll ask you some more physiology-based questions. Well, this is more anatomy. What are the name of the eye muscles? Where are they attached? What do they do? So now I'm going to stop sharing this. So you just go through it and you'll just type in any of these blanks here. If they have underlines, of course, the underlines are going to get mixed up, but it'll be okay. Just try not to completely mess up the format. It'll make it too hard for me to grade. So you can delete the underlines. I may try to do that and post it for anybody that hasn't printed this or saved this yet. So let me stop sharing this and then let me pull you up in Visible Body to help you understand what some of this where to go information means. So you can use some of the, or get some of the information out of the Human Anatomy Atlas, but that will only get you the anatomy part for the most. More of the physiology is in the Anatomy and Physiology app. Ugh, crashing computers, such a pain. So let's try this again. Okay, so unfortunately it crashed, logged me out of Visible Bodies. Give me 1 second here. So you're aware that this program is slower to open up. So when we get in here, if you've gone into the Anatomy and Physiology app before, there's a list of different topics on the left-hand side. So we're going to go in here and we're going to scroll down and we're looking for the nervous and special senses sections. Then if you scroll down, you'll get to the special senses, and it starts out with olfaction. So smell and taste, which we don't have time to do. Then we'll go down and it has the eye information. The first one has to do with where the eye is located in the skull, and then you go into the next sections and it goes along with those pictures in the lab handout that you have to turn in. So depending on what structures are asking you about, you can go into the different parts. Of course, you can always click forward and backwards into the different sections, depending on what you need. But if it asks you about a certain thing, like the lacrimal sac, what you need, there's some information on the side here, but the more detailed information is the stuff that's actually in this little book icon here. I mean, you can go through and have it read it to you and help you pronounce some of this stuff. But this book icon here has much more detailed information, especially the physiology. So one of the questions it asks you further down on that assignment is about tears. What are tears made out of? They contain salts, mucus, and protective enzymes. So depending on what structure it's asking you about, just find it, and the book icon tells you what it does, why it's there. So we're actually trying to get you to read and understand a little bit more about the physiology of these structures. So we'll throw in the anatomy and bring in the physiology. Things like melanin. Your iris and part of your other parts of your body have melanin. Why is it there? It gives it color. It helps, it's black because it helps absorb light, that kind of information. So most everything is in this app somewhere, or the Human Anatomy Atlas app. Let's go back real quick. There are some, or I guess I can hurry and scroll ahead. So there is a quick video also, so we'll go through the different views real quick. So depending on what they ask you, if they ask you about nervous parts, the rods or cones, there is also this video that summarizes again how vision works. All the information that's in the video is here on the side. Then there's a question that asks you about farsight and nearsightedness. You'll need to use this information here. And then it also asks you about the optic nerve and how it transmits signals to the brain. There's not actually a question on there. Unfortunately, I probably should have added it because it was not in the original, but it is kind of interesting to understand how the nerve impulses pass from both sides. So you see the color here shows you that each eye is getting some input from both sides of the brain, which helps bring the vision together and helps give us our binocular vision. So that's if you ever damage an eye, you know that it's much more difficult to orient yourself. So that's the vision material. Shouldn't be too bad. It is going to take you an hour, give or take, if you focus and get through it. So you can always do some questions and come back. Between the two handouts, a couple hours to get through everything. And the material that you're working on, please try to make sure you're actually understanding it, because there will be some questions on this information on the final exam next week. Any questions on that? So we're just trying to add together the information that we learned on the eye models and the cochlea slide and that kind of stuff. And when we do here in a second hearing, which is cochlea in the ear, that information and actually making it hopefully make more sense when you actually can put the function together with the anatomy. We're so used to pretty much just doing anatomy. It's nice to actually be able to bring in some of the, okay, what is it doing. And unfortunately, in lecture, you may not get to spatial senses because you tend to run out of time, and that's one that's less important than the nervous system or the bones or the muscles, so it can be removed and not come back to, I'm afraid. So this may be all you get on special senses in A&P until you get to professional school. So now let's move on and let's work on hearing. It's like I see the tab up there, but unfortunately there it is. I was like, it's not giving me a whole lot of help here looking for it. I finally did it.
Oh, I'd like you to think about how I'm doing this right now. Not why I'm doing it, because of course I'm doing it because I like music and I like science and I like to do both those things at the same time. But how can I play music? How can I be hearing it right now? And how can I walk around and play my guitar at the same time without falling on my face? And what is even sound anyway? These are all good questions. Let's start with the last one first. The basic answer to what is sound goes like this. Sounds create vibrations in the air that beat against the eardrum, which pushes a series of tiny bones that move internal fluid against a membrane that triggers tiny hair cells, which aren't actually hairs, that stimulate neurons, which in turn send action potentials to the brain, which interprets them as sound. But there's a lot more to our ears than allowing us to experience the pleasure of birdsong or the pain of grindcore. The ear is often overlooked, but even more vital role is maintaining your equilibrium, and without that, you wouldn't be able to dance or strut, even stand up. And you definitely could not do this. At least not without throwing up. In order to really get to the nitty-gritty of how your ears pick up sound, you got to understand how sound works. The key to sound transmission is vibration. When I talk, my vocal folds vibrate. When I slap this tabletop or strum a guitar, those vibrations cause air particles to vibrate too, initiating sound waves that carry the vibration through the air. So this sounds different than this, because different vibrating objects produce differently shaped sound waves. A sound's frequency is the number of waves that pass a certain point at a given time. A high-pitched noise is the result of shorter waves moving in and out more quickly, while fewer, slower fluctuations result in a lower pitch. How loud a sound registers depends on the wave's amplitude, or the difference between the high and low pressures created in the air by that sound wave. Now, in order for you to pick up and identify sounds from beeping to barking to Beyonce, sound waves have to reach the part of the ear where those frequencies and air pressure fluctuations can register and be converted into signals that the brain can understand. So once again, it all boils down to action potentials. But how does sound get in there? Your ear is divided into three major areas: the external, middle, and inner ear. The external and middle ear are only involved with hearing, while the complex hidden inner ear is key to both hearing and maintaining your equilibrium. So the pinna, or auricle, is the part that you can see and wiggle and grab or festoon with an earring. It's made up of elastic cartilage covered in skin, and its main function is to catch sound waves and pass them along deeper into the ear. Once a sound is caught, it's funneled down into the external acoustic meatus, or auditory canal, and toward your middle and inner ear. Sound waves traveling down the auditory canal eventually collide with the tympanic membrane, which you probably know as the eardrum. This ultra-sensitive, translucent, and slightly cone-shaped membrane of connective tissue is the boundary between the external and middle ear. When the sweet sound waves of your favorite jam collide with the eardrum, they push it back and forth, making it vibrate, so it can pass those vibrations along to the tiny bones in the middle ear. Now, the middle ear, also called the tympanic cavity, is the relay station between the outer and inner ear. Its main job is to amplify those sound waves so that they're stronger when they enter the inner ear. And it's got to amplify them because the inner ear moves sound through a special fluid, not through air. And if you've ever gone swimming, you know that moving through a liquid can be a lot harder than moving through air. The tympanic cavity focuses the pressure of sound waves so that they're strong enough to move the fluid in the inner ear. And it does this using the auditory ossicles, a trio of the smallest and most awesomely named bones in the human body, the malleus, incus, and stapes, commonly known as the hammer, anvil, and stirrup. One end of the malleus connects to the inner eardrum and moves moves back and forth when the drum vibrates. The other end is attached to the incus, which is also connected to the stapes. Together they form a kind of chain that conducts eardrum vibrations over to another membrane, the superior oval window, where they set that fluid in the inner ear into motion. The inner ear is where things get a little complicated, but interesting and also kind of mysterious. With some of the most complicated anatomy in your entire body, it's no wonder it's known as the labyrinth. This tiny, complex maze of structures is safely buried deep inside your head because it's got two really important jobs to do. One, turn those physical vibrations into electrical impulses the brain can identify as sounds. And two, help maintain your equilibrium so you are continually aware of which way is up and down, which seems like a simple thing, but it is very important. To do this, the labyrinth actually needs two layers. The bony labyrinth, which is the big fluid-filled system of wavy wormholes, and the membranous labyrinth, a continuous series of sacs and ducts inside the bony labyrinth that basically follows its shape. Now, the hearing function of the labyrinth is housed in the easy-to-spot structure that's shaped like a snail's shell, the cochlea. If you could unspool this little snail shell and cut it in a cross-section, you'd see that the cochlea consists of three main chambers that run all the way through it, separated by sensitive membranes. The most important one, at least for our purposes, is the basilar membrane, a stiff band of tissue that runs alongside that middle fluid-filled chamber. It's capable of reading every single sound within the range of human hearing and communicating it immediately to the nervous system, because right smack on top of it is another long fixture that's riddled with special sensory cells and nerve cells called the organ of Corti. So when your cute little ossicle bones start sending pressure waves up the inner fluid, they cause certain sections of basilar membrane to vibrate back and forth. This membrane is covered in more than 20,000 fibers, and they get longer the farther down the membrane you go. Kind of like a harp with many, many strings, the fibers near the base of the cochlea are short and stiff, while those at the end are longer and looser. And just like harp strings, the fibers resonate at different frequencies. More specifically, different parts of the membrane vibrate depending on the pitch of the sound coming through. So the part of the membrane with the short fibers vibrates in response to high frequency pressure, and the areas with the longer fibers resonate with lower frequency waves. This means that all of the sounds you hear and how you recognize them comes down to precisely what little section of this membrane is vibrating at any given time. If it's vibrating near the base, then you're hearing a high frequency sound. If it's shaking at the end, it's a low noise. But of course, nothing's getting heard until something tells the brain what's going on, and the transduction of sound begins when part of the membrane moves and the fibers there tickle the neighboring organ of Corti. This organ is riddled with so-called hair cells, each of which has a tiny hair-like structure sticking out of it, and when one is triggered, it opens up mechanically gated sodium channels. That influx of sodium then generates graded potentials, which might lead to action potentials, and now your nervous system knows what's going on. Those electrical impulses travel from the organ of Corti along the cochlear nerve and up the auditory pathway to the cerebral cortex. But the information that the brain gets is more than just like, hey, listen up. The brain can detect the pitch of a sound based solely on the location of the hair cells that are being triggered. And louder sounds move the hair cells more, which generates bigger graded potentials, which in turn generate more frequent action potentials. So the cerebral cortex interprets all those signals and also plugs them into stored memories and experiences so it can finally say, oh, that's a chickadee, or a knock at the door, or the slow burn of an 80s saxophone solo, or whatever. So that's how you hear. But we're not done with you yet. We got to talk about equilibrium. The way we maintain our balance works in a similar way to the way we hear, but instead of using the cochlea, it uses another squiggly structure in the labyrinth that looks like it's straight out of an alien movie, a series of sacs and canals called the vestibular apparatus. This setup also uses a combination of fluid and sensory hair cells, but this time the fluid is controlled not by sound waves, but by the movement of your head. The most ingenious parts of this structure are three semicircular canals, which all sit in the sagittal, frontal, and transverse planes. Based on the movement of fluid inside of them, each canal can detect a different type of head rotation, like side to side, and up and down, and tilting, respectively. And every one of the canals widens at its base into sac-like structures called the utricle and saccule, which are full of hair cells that sense the motion of the fluid. So by reading the fluid's movement in each of the canals, these cells can give the brain information about the acceleration of the head. So if I move my head like this, because I'm like super into my jam, that fluid moves and stimulates hair cells that read up and down head movement, which then send action potentials along the acoustic nerve to my brain, where it processes the fact that I'm bobbing my head. And just just as your brain interprets the pitch and volume of a sound by both where particular hair cells are firing in the cochlea and how frequent those action potentials are coming in, so too does it use the location of hair cells in the vestibular apparatus to detect which direction my head is moving through space and the frequency of those action potentials to detect how quickly my head is accelerating. But things can get messy. Doing stuff like spinning on a chair or sitting on a rocky boat can make you sick because it creates a sensory conflict. In the case of me spinning around in my chair, the hair cells in my vestibular apparatus are firing because of all that inner ear fluid sloshing around, but the sensory receptors in my spine and joints tell my brain that I'm sitting still. On a rocking boat, my vestibular senses say I'm moving up and down, but if I'm looking at the deck, my eyes are telling my brain that I'm sitting still. The disconnect between those two types of movement, by the way, is why we get motion sickness. It doesn't take long for my brain to get confused and then mad enough at me to make me barf. And I'm sorry that we're ending with barf, but
Okie dokie. Well, let's go through now and just pull up the hearing handout you have to do. So just like with the vision handout, you're going to go through and put your name on here. When you turn it in, make sure that you label it with your name, and then if it's hearing or vision, all the gaps you will fill in for information, so you just click on it and type it in. Trying to do your best not to completely skew stuff so I can't understand what you're writing about. It's like you can be picky about your formatting and tab it in, or just leave it as it is. It's fine. Just go in here and work on filling in all the information. The key thing is to look for things like the tympanic or tympanic membrane, or the external part of the ear, which is your auricle or your pinna. Those kind of terminologies, because when you get into that visible body, those are the kind of words that are going to help you find a structure to click on, which then you can go to the book icon and find. So just go through and fill all this in. On the vision handout, sorry, I'm going fast, double check. There are 2 activities they ask you to do where they say to use a partner. If you do not live with anybody that you can partner up with, they can be done by yourself. They're not as accurate, but you can still get the gist of what's going on if you do it by yourself. I don't think there is anything like that in hearing, because you would have to have some kind of device that's generally not something to come across in your house. We use tuning forks, but those generally you don't have laying around your house. You could probably use some kind of computer app for that, but we don't ask you to do any hearing tests on yourself. There is kind of a interesting app though that plays different frequencies and then gives you an estimate of how good your hearing is. So there are fun free apps out there that do that kind of stuff. But just keep going through here, all the gaps, fill in the information. They tell you, I try to tell you what section stuff come from, but the key thing is look at the pictures. As we go along, we went from the outside of the ear, and now we're getting deep into the inner cavity, and that goes along with the visible body. So let me stop sharing this and go to visible body. So we stopped here, and you could go all the way back to the beginning if you wanted to. Otherwise, depending on if you come back and forth, you would click down and you would look for the hearing stuff. Otherwise, I'm just attempting to click on because the hearing stuff comes right after. So it asks you questions about what's the function of this external ear. So you've got your book tab that helps you tell you that kind of information, like the external acoustic meatus. What is it there for? How does it function? What's its shape? Why is its shape good? So he showed you this in the movie. One of the questions asks about why do you have this pinna on the outside here? How is its shape important? And he talked about its job is to kind of the sound hits here and it kind of gets pushed or funneled down into your smaller ear cavity here. So there's a bunch of those kind of questions. So just go through, find the parts that are relevant to the topics it's asking you about. It'll start at the outside and work their way all the way into the inner ear here. Can't do as much manipulation as far as hiding and removing stuff. It's froze. There we go. But you can still open the book tags for all this kind of stuff. We can't tag stuff like we can in the atlas. That you will have to wait for them to manipulate stuff. You can't like pull things apart or remove stuff, which is the upside to using the Human Anatomy Atlas. Is just not as detailed information. So if you like to use that, that's fine. Just realize that the book tabs don't generally have quite as much information in it, and you may have to come back and use this program in addition. You will have to come back to this at least for the videos, because that's physiology. So the information, so you can listen to the video and watch what's going on, of course, because that visual input will help you learn it. But then the words, if you wanted to search through for some of the information, instead of trying to interpret how things are spelled or pronounced, you can go through and you can see the words actually typed out. So everything that's said in the video is typed out on the side here. I did not add equilibrium on there. I still want you to be familiar with, of course, the structures and look through this information, but there are no direct questions on the hearing handouts. It would have to be a equilibrium balance handout. So that this information is not on the handout, but still be familiar with it, please. I mean, there's not much to it. It's just a little bit of that inner stuff going on there. So I'm going to stop sharing that, and that is it. So we're officially done with all the new material once you get through this. If you will go into the chat box and please type that you're here. Other than that, is there any other questions? So there's no meetup on Monday. There's a meetup on Tuesday and Wednesday. You have to pop into at least one of those if you want your 5 points for attendance next week. The goal is just to ask questions, pull up any of the PowerPoint presentations, any of those other kinds of stuff, and help you with anything that you don't understand or you're having trouble with for the exam, which starts at would be 12 a.m. on Thursday morning and goes until 11:59, give or take, midnight on Thursday. So it's 24 hours only this time. So if you have any conflicts, let me know ASAP, because unfortunately we have to get grades in soon after that. So are there any other questions? Any questions at all? You can send me an email, but for this handout, it's visual. So we're probably going to have to make an appointment if you need to see where some of these answers come from. And I would just go into that course room in Blackboard Collaborate Ultra, and we'll just make an appointment and we'll meet in there. I can do that, of course, also for reviewing for the final. But the goal with the meetups on Tuesday and Wednesday is to try to eliminate trying to squeeze questions in at random times that I'll be there and you can ask questions. So if there's nothing else, I'm going to stop recording.